I want to thank the organizers, the board, the medical board, and patients' families for the opportunity to tell you a little bit about the work that we are doing in Philadelphia. Now, I am not a rheumatologist by training. I'm a pediatric pulmonologist, so I'm a lung specialist for children, but my PhD gave me training in molecular biology and genetics. I was also with Decode Genetics in Iceland, which is so really a large-scale population genomics enterprise. We were basically involved with genotyping and genomically characterizing the entire Icelandic nation, which was quite an enterprise. And I came back to Philadelphia to found the Center for Applied Genomics in 2006. So I wanted to give you a little bit of background of the center and then tell you about one success story, actually of a disease in my area. It's a lymphatic disorder, because the approach that we are taking to JM is very, very comparable. So next slide, please. So the field of genetics and genomics is actually very exciting today. And as you know, I mean, there is actually stem cell therapy and induced stem cell therapies have actually been successful treating cancers, treating metabolic disorders. And one particular therapy that came out of Children's Hospital of Philadelphia is this gene therapy for Leber congenital amaurosis. So this is the retinitis pigmentosa gene number 65 that has mutations. And actually, if you put a normal gene into an adenoviral vector, you can inject that into the eye. And children who are born blind, they can see. So it's quite remarkable. Targeted T-cell or CAR T-cell that Lisa mentioned earlier, this therapy came out of the University of Pennsylvania and CHOP. First trials were done at CHOP. And this is now pioneering therapy for leukemia and some other hematological disorders that are resistant to other therapies. But this is now entering into other disease areas, other solid tumors and other diseases like autoimmune disease. So quite remarkable. And then the gene editing opportunities. I mean, this is sort of really the future of therapy, because I mean you can actually get DNA circulated from blood from pregnant mothers. As you know, you can diagnose any known condition, and you can actually apply gene editing. This is done in animal studies today, and it's just a matter of time when this sort of advances more into therapies. Single-cell sequencing gives us also the opportunities now to really specifically assess what's happening at a single-cell level. Because if you take, for example, a joint or skin, there are multiple cell types, and we can now look at these cells individually, look at the expression and function of all genes at the individual cellular level. We have one of the pioneering programs actually at CHOP, at the Center for Applied Genomics for this. So next slide. Center for Applied Genomics is actually one of the center of emphasis at CHOP. There are 10 such centers. So CHOP is the Children's Hospital of the University of Pennsylvania. So we work very closely across with Penn investigators. About half of our collaborations are actually with Penn investigators. And CAG is actually by far the largest of the centers. We have about 75 employees in our center. Next slide. So the founded in 2006, the goal was to basically recruit families and patients from CHOP and establish a large-scale biobank. We've actually now recruited about 130,000 children and family members, so unique families. We have about 450,000 samples from all of the collaborative efforts that we have worldwide. As you know, the team from CAG has been here at past several meetings. They are here now across the pond collecting samples, and we are immensely grateful for your interest and willingness to participate in these studies, because this is really the critical assets that allows us to make discoveries. So the electronic medical records that we utilize, they're not perfect, but we have sort of come up with algorithms to make them more valuable, and we really work across essentially any pediatric disease, both common and rare. We have a special team on this project that Charlie Cow leads, who was here and presented last year, and we hope to have an opportunity to present to you in the future some really novel discoveries coming from the efforts of sample collection and history taking from here. Next slide. So just to emphasize that, as was brought up before, if a disease presents itself at an early stage, the genetic component is usually so much more robust, so much stronger, that it's much easier to find that. And some of the diseases that present at early age, even though they're complex, they're essentially monogenic. So we have, we've published actually over 700 papers from the CAG Center. And we have often identified genes and genetic factors that are critically important for disease pathogenesis with a fraction of the sample size that, for example, adult studies have. And that's because they are not molded and derailed by various environmental influences and other things. So it's really a huge advantage to study children. The younger, the stronger the data. Next slide. And just to emphasize here, sort of, this is sort of a setup for the past decade or so in terms of studies that we've done, that we are now a CLIA certified lab, which means that we can actually do clinical testing, so we can get samples from anywhere, actually in the US or in the world for that matter, and do a clinical test. And we are collecting samples that sort of, we use to validate the assays that we intend to use for this type of discovery work that we are doing for JM. Next slide. and this is another quickie, just an overview of sort of the type of samples. You can see that autoimmune inflammatory samples, we have 27,000. This is also extremely valuable when we have a rare condition such as JM, because we have the comparativeness of multiple other, we have over 30 different autoimmune diseases in our collection that we can use as a comparison and reference, plus all the healthy controls that you need in order to sort of, make discovery. Next slide. And the Biobank, Charlie has probably shown this slide before. I mean, this is a sort of a robotic-based facility that we have, and we store all of our samples in a biorepository setting. This particular robot can hold up to 10 million samples in plates. So we still have, plenty of room to store and collect and share samples from that. Now, we always isolate cells because it's critically important to be able to subsequently study RNA and protein to sort of figure out what these genetic mutations that we identify do. And we also store plasma, so we can go and measure any sort of protein level or biomarker that we may find. So next slide. And again, I mean, the genome is complex. It's about 6 billion base pairs, 3 billion coming from each parent. There are differences, if you will. We call them polymorphisms that happen every 200 to 300 base pairs. And, so there are millions of them. So the key effort here is to find the ones that are disease-specific, disease-relevant, and utilize them to identify patients who have certain biological pathways where a specific therapy can be utilized based on the genetic underpinnings. That's what we try to do. Next slide. And again, big data is critically important. I mean, you've sort of heard the concept of big data. Now, with the biobank that we have with all the information, we get updated records from our patients at CHOP, and we recontact with these families as well. We can bring patients back for if we have, opportunities to intervene and treat or recommend different treatments. Next one. So this is a project that I'll go quickly over this before I get into the JM topic, but because it's sort of an interesting, there are two cases here. Lymphatic conditions, they are similar to JM. There are multiple types of lymphatic conditions that differ, but what we have found is that there are certain mutations that cluster in on certain biological pathways that actually work across all of them. The mTOR pathway, some of you may be familiar with a drug which is called rapamycin, and is sort of anti-inflammatory drug which is utilized to treat cancers and various inflammatory disorders. That pathway had actually been sort of identified in the context of lymphatic disorders, where the certain kinases, we call them PIC kinases, and the mTOR have been identified with acquired mutations, so we call them somatic. They're not born with these mutations, but they acquire them. The drug rapamycin or sirolimus has been utilized to treat them. Next slide. So this was actually a CHOP faculty who had came to me a few years ago and had this 4-generation pedigree, which is basically a dominant inheritance of this very, very rare condition. And her son was affected sort of most significantly of all of these individuals in the family. And he had multiple TAPs, cauterizations, occlusive procedures to try to sort of, block the disease. Some of the other family members had less of an involvement, but it was unknown what this sort of the causes were here. Next one. And here you can see the CT scan with a fair amount of fluid. I don't know if this, no, it probably doesn't show. On the CT scan, and you can see that the lymphatic vessels are all tortuous and sort of twisted, and this patient was leaking fluid all over the belly, chest, pericardium, and elsewhere. Next one. And we found a mutation which is an insert that sort of resulted in shutting down the function of this gene, which is called Ephrin B4. And Ephrin B4 is important if you go to the next slide, because this gene is critically involved in the development of both veins and of the lymphatic vessels that develop out of the veins. So you can see it sits here sort of on the venous side of the diagram. Next one. And we took them, this observation, and we found the homologue of this gene in a zebrafish. And we created this same mutation, actually, because this was loss of function. We could just knock out the gene from the zebrafish. And there was all kinds of misbranching and dysregulation that happened in the fish. Next one. And when we looked at there was certain kinases, this P70 phosphokinase that was highly upregulated, but the mTOR pathway was sort of upregulated with the loss of this Ephrin B4 gene. And we could actually take the fish and treat the fish with rapamycin and with PIK3 kinase inhibitors and essentially reverse it, not totally completely, but in a significant way. We did the same thing with cells from the patient, and we reversed the abnormal cellular proliferation in this individual. Next one. And this sort of just highlights that, there's a gene discovery that causes loss of function of this pathway. We put it in a fish, we create a phenotype in the fish, we treat the phenotype, and here we have a treatment which subsequently we applied to this family with great success. Next case is actually even more intriguing, if you go to the next slide, because this was a patient who had been on this drug with, and pushing doses and was just getting significant side effects and had no benefit from it at all. And this, you can see the pericardium here is filled with fluid, his belly is all swollen, and it's a very tortuous sort of, lymphatic system. And no mutations had been found in this individual. If you go to the next one. So we found actually mutation in a totally new pathway, which is called ERK, or extracellular signaling sort of, it's a MAP kinase RAS signaling pathway. And there were 2 patients actually who had this mutation with the same phenotypes. It was very unlikely that this was by chance, that this was not the cause of this. If you go to the next one, you see that this pathway here on the right-hand side is actually very separate from the mTOR pathway. So the kid who had been treated with sirolimus, he had no benefit because the mutation was on the other side of it. If you go to the next one, and here, you know, the protein upregulation, there was no change in this P70 kinase, but the MEK and ERK, they were highly, highly upregulated in this individual. And none of the sort of molecules in the other kinase pathway were affected. Go to the next one. And here you see effects of therapy, that a drug that blocks this particular pathway, it completely reverses this abnormality in the cells, and basically patient material. And we took it subsequently into the fish. If you go to the next one. This is actually a sort of so-called sprouting assay, that it's a spheroid sort of culture of cells from the endothelium, and they grow in different directions. So you could see that you normalize this completely with a drug called trametinib. It inhibits this pathway. Next one. Same holds true with all of the cellular changes that are very abnormal, vascular endothelial molecules. They completely normalize. Next one. Actin filament that are all derailed in the system, in the cells, similarly reverse with therapy. Go to the next one. And this is the fish data. You may not see this well, but there was a very significant misbranching and abnormality in the fish. And if you go to the next one, this got essentially completely reversed with therapy. So this is sort of, you know, if you go to the next slide, this is sort of what we are hoping to do. This patient was actually dying in the hospital. His lung function was down to 23, and you can see his lymphatic system here on the figures below his lung function test. And a few months into therapy, he basically is now biking, you know, all daily activities, running, and cured. And you know, he was basically admitted like every couple of weeks for surgical procedures because there was nothing working for him. So go to the next one. We now actually have, and here you can see that his lymphangiogram actually essentially is now normal. So he just normalized his lymphatic system by this treatment, which is quite, I've never ever seen any sort of remodeling of that type happen for any other tissue before. So it's quite remarkable. If you go to the next one, we actually have now found 10 other genes since we found this one. This took about 100 patients to find, and now we have 3 patients on therapy, and they have all had transformative response. And so this is what we are hoping, if you go to the next one, hoping to do for JM, sort of applying this particular model and test the mutations in models and cells, reverse them with medications, and then get IRB and FDA approval for compassionate use of new novel drugs. We submitted a frontier program on this that we got awarded, and so now I'm going to shift gears and tell you about the status of the JM work. If you go to the next slide. And the next one, so Charlie, next one, Charlie has actually led the development of an interferon assay. We heard about interferon, type 1 interferon and the role this morning, essentially from all of the speakers, but there are 13 of these interferon alpha genes, 2 beta, and then kappa and theta that are found in humans. And the alpha receptors, R1 and 2, they are the ones that respond to interferon 1s. And this is an antiviral sort of response mechanism and potent sort of inducer of inflammation, including upregulates all of the MHC class 1 and class 2 molecules. Next one. And you saw this similar slide from Lisa earlier this morning, that this is sort of, the point here is that the interferon sort of signaling that goes through the JAK-STAT signaling has multiple phosphorylation sites on the way down through the nucleus, and there are multiple avenues to intervene at this level. So Lisa mentioned the monoclonal antibodies, tofacitinib, Bara, and Ruxo, that have been developed. There are interferon receptor and interferon protein blockers available. SOX1 is actually a drug that we are developing at CHOP. Which is a negative regulator and may have broader impact than the JAK2. All of these interferon genes are very robust, so they sort of present good biomarkers for testing. Next slide. So this is a paper on lupus, and Lisa similarly showed something similar before, so I'm not going to dwell on it. Next slide. Just showing that there are, a few hundred genes that are highly robustly responding to the interferon and can sort of be interpreted as a signature for interferon. And if you go to the next one, there is actually a little further up. There's actually history, quite a bit of work that has been done in this area, as Lisa and Anne and others reviewed. Muscle biopsy, blood cells, juvenile myositis patients have been sort of shown to have interferon signature, but there hasn't been an assay developed to sort of formally address this. Seen in dermatomyositis, juvenile myositis, polymyositis, and particularly in patients who have anti-Jo1 autoantibodies. But what is interesting is that the intensity of this seems to correlate, at least there is suggestive data that it correlates with the response to therapy or the disease activating status. So JAK inhibitors are one type of drugs that you would think might be of value if you have a high profile for this signaling pathway. Next one. And why should we do this? Well, I mean, it's a measure of one of the way of sort of measuring disease activity. It's another way to identify a subset of patients who are maybe primarily driven by this signaling pathway, and therefore intervention at this pathway could be a powerful way to treat them. And again, JAK inhibitors, interferon blockers, or other drugs that work in the pathway would be something that we will now study in the cell-based assays, looking at the different mutations, particularly in the patients who have these profiles highly upregulated. Next one. So Charlie has led this assay development. So there are 32 response genes that we took, and we tested them with this nanostring technology in 19 patients. So you can see that there is different activation status among these patients. These were all patients that we have collected from the various conferences and meetings from the CureJM families. And then, there were 5 controls that we utilized for this first sort of panel. And if you go to the next slide, you can see that the controls are relatively low. But there is a handful of patients who are actually very, very significantly upregulated. And most of the patients actually don't have a signature for interferon type 1. So this is actually important because if we look at this, actually the patients with the active disease, they are actually enriched in that upper right signature profile. And this is sort of, looking at only 16 of the most relevant genes in the pathway, which would be the assay that we take forward into development. Next one, and again, patients on the right side, controls on the left, and you can visibly see the difference. If you go to the next one, this patient actually is a CHOP patient that we have interest in approaching. We've already spoken with Ed Behrens, who is chief of rheumatology at CHOP, and we will be seeing if this individual who has actually significantly active disease would be willing to consider going on a drug in a low dose that sort of, is not having a lot of side effects at that level. Next one. And this is sort of a pilot that we will then scale and expand. We are going to do RNA sequencing, so we're going to test the sequencing signal in all of the genes in the human genome to see if they're any better. We can fine-tune this panel, but then, we want to validate this panel and make this available as an assay. This is actually an inexpensive panel, can be marketed for a very low price, but could be very valuable to pick out the patients who have this pathway involved, which is maybe about 20% of JM patients. And again, allows us then to explore new therapies, of which there are multiple in this sort of pathway. So next slide. Update on the sequencing work that we have been doing on the samples that we've collected from the various sites. So the exome, as you know, is just a small subset of the whole genome, but it is the sequence of the coding sequences of the entire genome. So that's typically where most things are happening, and it's just much cheaper and easier to sequence the exome than it is to sequence the whole genome. So this is what we have taken on now and completed about 70 plus samples, looking for rare variants that are absent in our control subjects or in public databases that are available. So if they're present and enriched in patients with JM, they may have disease-causing effects. So family history, family records are critically important for this purpose. Next one. And again, this is just to show, Charlie actually presented on this last year, and now with the samples we are collecting and will be sequencing, we will be validating whether the discoveries that we made from the various biological pathways that he showed hold up in this set, and we see enrichment in an independent data set. So that's going to be sort of one of the evaluation that we do. The other is to look at the individual pedigrees, particularly pedigrees where there are complex sort of combinations of different diseases where parents may be affected and where there is more than one child affected. On the left pedigree you see here, parents are healthy, other siblings are healthy. This is very likely to be what's called a de novo mutation, that is sort of spontaneous mutation in the from the germline in the child. But on the right-hand side, and if you put up the next slide, you can see that it's exceedingly likely that there is an inheritance factor from the mother in that instance. And families with multiple people and multiple potentially different autoimmune diseases are most informative actually for this type of work. Next one. So this is just a couple of families that we've looked into in more detail. Both parents here are affected with autoimmune disease, and both children have JM. In this instance, and it's actually interesting, because the catalytic subunit of the PIK3 is actually one of the genes that is transmitted here from the mother in this particular instance. That's the same pathway as I showed you for the lymphatic. So there are multiple drugs, and those kids we have treated for the lymphatics, the cells are so much more sensitive than the cancer cells that we can use a fraction of the dose that have essentially no side effects at all. So that would be the hope, for example, when we explore this. There are a couple of other things. There are these PPR, peroxisome transcription factor, that is involved in mitochondrial. So there may be more. This is not a single gene disorder. It's a complex disease. But if you knock out one of the pathway out of many interacting pathways, you may do enough to suppress the therapy. There's another cytotoxic T-cell activating gene here, which comes from the father in this instance, which is also of interest and with respect to biology. We are going to be testing all of these now in the cells from this family. Next one. And here is another even more complex family where the mother has an autoimmune disease, father has some form of inflammatory condition, but it's not confirmed. One child with JM, another with some other autoimmune condition, and the third one is not normal, but with an inflammatory state. So there are a couple of sort of, after all of the filtering and analysis, genes that come out. This is the NFA-TC3. It's a nuclear factor transcription factor, which is actually very important for calcineurin T-cell activation, and there are drugs available for this. And the second one similarly activates the phosphoinositol pathway, and there's a drug involving IL-12, IL-23, which we will test and figure out if that has any impact in this particular, in terms of reversing the pathology like we saw in the lymphatic system. Next one. So the future direction here, functional profiles on all of these in the cells. We can actually take the cells and generate iPS cells, differentiate them into any muscle or cells of interest, so we can test sort of more relevant cell for the context of these experiments. There are more individuals than this family that we found with the PIK3, which is the overlapping pathway I showed you with the lymphatic disorders, and then the other genes that are highly relevant, potentially relevant candidates. They all have drugs, which is important. And so we will be, as I said, finalizing the sequencing from the new collection. There will be about 200 families in total coming from the concerted effort of these recruitments, which is quite impressive. And we will be forming sort of association and discovery efforts on new pathways and testing them with the notion of submitting a frontier grant at CHOP. And we hopefully will have one of the patients already in, which will sort of be a ticket of a successful grant. Next slide. And again, all of this is, the biobank is the critical thing here. So the sample collection, that's the foundation. The technology changes all the time, but the samples are there, and these samples are, we have cells, we have DNA, we can share all of this with anybody here in the audience for future studies. And with that, next slide, multiple people to thank. Charlie Cow has done most of this work with his team, and the clinical recruitment staff who is currently across the pond, as I said, and hopefully will be bringing another successful collections with that. I want to thank you for the opportunity to present. I am not listing funders here, but we are immensely grateful for the contribution from the Cure JM Foundation for this. Thank you.